All right, so we'll go ahead and get started. Hopefully everybody clearly today. Thank you for being here for the speaker series. We're really happy to have Dr. KC Lee back with us again. Dr. Lee is professor and chair of speech and hearing sciences at the University of Washington. He got his bachelor's degree in electrical engineering from the University of New South Wales, and his doctorate from Harvard MIT Health Sciences. Dr. Lee's diverse research focuses on developing multimodal imaging techniques, investigate the cortical network involved in auditory scene analysis and attention, especially through designing novel behavioral paradigms that bridge the gap between acoustics and research. So we have a really interesting talk lined up. Turn it over to Dr. Lee. Thank you.
Thank you. Thank you, Roland. And you can hear me properly?
You sound great.
Okay, great. Thanks. All right. So thank you for the invitation to talk again. I'm glad that I had the opportunity to speak to you 2 weeks ago, and today it will be on the topic of my research. First, I want to acknowledge on behalf of University of Washington. I want to begin my talk by acknowledging the coast leadership peoples of the land on which we do our work, the land which touches the shared waters of all tribes and bands within the Suquamish, Tulalip, and Muckleshoot nations. So the roadmap of my talk will begin with asking what the cocktail party problem is. I think you've all experienced it one time or the other, probably less so during COVID, but it's coming back. And then I'll talk about the general behavioral and neuroimaging approach, and then investigate what the cortical dynamics of auditory attention is sort of the focus of our lab. But I want to also link the oculomotor circuitry, as in how you move your eyes, into the auditory attentional network, and then leave you with some future directions of where we want to go next. So let's start with the cocktail party problem. So this is the normal social setting when we converse in a social setting. Normally, we just don't have one speaker talking at a time. We often have multiple speakers talking all at the same time, and your task is really following a conversation with one speaker while actually trying to ignore the other speakers, or else it would be too overwhelming. And so this is coined as the cocktail party problem back in 1953 by E. Colin Cherry. And so we have to pass the scene, the auditory scene, and it's slightly different from vision. So in a visual analogy, if you're staring at the screen and I'm on it, and you can see the edge of that screen, the edge of that screen demarcate the edge of the object of the screen. But in audition, we call this more like a transparent scene. So you have multiple objects that are overlaid on top of each other. We rely on cues to help segregate these objects. So here I'm using color as a way for you to see the equivalent of how cues can segregate the scene better. Now, attention can help you specifically promote one set of objects using these cues, and you can switch attention to one set of cues to another, thereby switching attention to different objects. And if you know, I think we share this in common, Huskies, it's a primed word both at UW and at UConn, and here Huskies is also a, priming also works in terms of scene segregation. So let's look at what type of cues that we use for grouping and selective attention. So what are the auditory cues that we tend to use? Well, there's timbre. So if you're listening to the orchestra, you can try to segregate out the string section versus the woodwind, and you can maybe concentrate specifically to the violins versus the flute. Intensity is another cue. I'm assuming that your projector has some low frequency hum, And so the difference between my voice and the low frequency hum, you can segregate sound that way. What we're going to concentrate on here in this talk is the spatial cues and pitch. And we're going to use pitch not as a cue per se, like studying pitch, but as a cue to contrast that is a non-spatial cue. Okay, so we talked about auditory spatial cues. What are the ways that we can perceive space in audition? So it turns out if you have a sound object coming from one side of the ear, because of time travels, the sound travels to your ipsilateral ear, the closer ear, slightly arriving at the sound arriving at the ipsilateral ear slightly earlier than the contralateral ear. So the interaural time difference is one way to produce this auditory spatial cues. And we are capable of noticing 10 microseconds of a difference, which is pretty cool. The head also acts as a head shadow for higher frequency. So the sound that is arriving to the ear that's closer to the sound object will receive that intensity slightly higher than the contralateral ear, and thereby having the interaural level difference. It's another cue for a spatial percept in audition. Turns out that the pinna also gives you a third spatial cues as well, the shape of the pinna. Okay, so now that we've talked about the basics of what we're talking about, let's go into the specific behavioral paradigm and the neuroimaging approach. So what we rely on is a modified Posner paradigm, in which we start by asking the subject to look at their fixation dot, and we cue them into listening, paying attention preemptively of a cue. So here, the arrow is pointing to the left. We ask the subject to maintain fixation, and we always have two competing sounds that's presenting to the subject. And here we have a spoken digit, 3 coming on the left, 2 coming on the right. And because the arrow was pointing to the left, the subject has to respond to 3, and we will mark that as a correct answer, and we will then process the brain data. Now, we can, of course, switch the arrow from pointing to left and right, and then instead we'll point to up and down. These two spoken digits also have a higher pitch or low pitch. So if we pointed the arrow to up, basically the subject is preparing for responding to the higher pitch sound. And when the sound comes on, the response now it would be 2, because the higher pitch talker basically spoke the digit 2 out loud. And so the subject can change the feature cue attention using these arrows. So we use magneto-encephalography with simultaneous EEG acquisition. On a separate day, have an anatomical scan using MR, and we co-register the census locations with the MRI, and with a bunch of math. Essentially, it's a minimum L2 norm solution with some penalty constraints. If you're interested in inverse imaging, ask away afterwards. But essentially, just some linear algebra. And what actually provides us, it's a way to map signals from the sensor space to the cortical space. And that's how we can get the cortical dynamics of auditory attention. So let's look at some MEG movies. So here, remember, we were giving you a visual cue, and of course, in the beginning, when the subjects are looking at the visual cues, the occipital cortex will receive waves of brain activities. During the time of fixation, you see that the precentral sulcus, or where the area that I just marked attention, has more activation, brain activation. Now, interestingly, fixation is also held in this sort of frontal eye fields area. So we're going to do contrast to see how attention is modulated depending on different cues. When the sound comes on at time 0, you see a wave of auditory activities. 100 milliseconds afterwards, it's the signature of the auditory response in the cortex. So that's how we generate these MEG movies. So then what we do is contrast the different cues. Now remember, here we asked the question, what happens if we point you to listen to space versus pitch cues even before the onset of the sound? Now, in fMRI, you can design these experiments to have ways to present the sound or not present the sound, and have trick ways to measure preparation, attention preparation. But fMRI, the time sampling is much slower, and also there's intrinsic noise of fMRI. So we, in auditory experiment, we really like the MEG approach and being able to have millisecond precision. So at the pre-stimulus intervals, even before the sound onset, we can see that the frontal eye fields area, pointed out on the left here, it's more activated compared to when the subject is paying attention to the pitch in the upcoming stimuli. This persists across even during the stimulus, so the differential activities in the frontal eye fields, it's sustained before the stimulus and during the stimulus. It turns out if you're paying attention to pitch, the STS, the superior sulcus, is more activated, but I'm not going to concentrate on that part as much here. Interestingly, frontal eye fields, if you think about how the terminology came about, it's when you zap the anatomy in the monkey, you actually elicit eye movements, right, in the frontal area. Now think about when you move your eyes when you're doing visual analysis, you're actually shifting spatial attention in vision. So it's interesting that the frontal eye fields is also involved in auditory space attention. So this is our first study. It was done back in when I was still a postdoc in MGH, actually. And so we decided to pursue this again. And I think one funny story here is that in the last figure that I showed you, I could only show it in the frontal eye fields on the left side and not on the right side. And at that time, I had one subject. It was me. I had to put myself into the scanner to produce preliminary data for the K99 grant. And I remember at that time I saw FEF differential activities, but it's only on the left and not the right, and I have no idea how to write the grant. That is not bilateral. And a funny story would be that, in the end, I included the left-hand side of the figure in the grant and said, due to space constraint, I'm only showing you with the left brain. And so now we want to do a follow-up study, seeing the frontal eye fields differentially involved in space. And one thing that, so I was trained as a psychoacoustician, and what drew me into neuroimaging is that we can ask the question, not what you're attending behaviorally, but we have a way to ask what happened to the sound that you were not attending to. And so here the task gets slightly changed a bit. We call this Old MacDonald's task. Basically, you have a bunch of sounds coming out, all the letters coming in, and you have to count how many E's and sounds like E, I, E, I, O. And in this case, you're cued either to listen to the left side or the right side. But we tag each of the sounds with a different amplitude modulation. So it sounds more like E, E, M, A, E, L, K. So that buzzing sound is the AM tag. And what that allows us to do is tune into that AM tag and look at the phase lock value and see not only what happens to the which part of the brain is locked to that the frequency, the tag frequency that the stream that you're attending to, but we can also dial to the stream that you're not attending to and ask what happens to the phase lock value at that carrier frequency. So this is a very engineering approach due to my electrical engineering background. And so what we saw is the following, and this is the work done with Barbara Sheen Cunningham and Harry Bharadwaj, when we were all back in Boston at that time. And so when we dial into the 35 hertz of the attended side, we see this again sustained PLV, the strong phase lock value in the frontal eye fields, again on the left side and not on the right. Now we can then tune to the unattended side. The carrier frequency would be 45 hertz, and that phase lock value is no longer there in the left side of frontal eye fields. You can play around with whether you're paying attention to the left side or the right side using frequency, carrier frequency of 35 hertz or 45 hertz. All of these, the strongest phase lock value happens on the left side of the frontal eye field. So the moral of the story, even though in a preliminary study where I can only find on the left and not right, and I wrote it as I can only show on the left side, even with follow-up studies, we can only show this on the left and not right. And I'm very much interested if anyone can give me some theoretical reasoning why that's on the left and right, and we're still chasing down that question. Okay, so switching gear, going to switching attention. Part of this is because as an engineer, I'm really interested in finding out whether you can capture sort of some neural markers of you switching of attention. Now, this comes from hearing aids design, right? So hearing aids it's hard to work in noisy environments. Basically, it amplifies what the computer considers as target, but really when you're conversing, you want to be able to switch attention from one speaker to another. So the question phrased here is, can we find out which region of the brain is more activated when the subject is switching spatial attention? So the modified task here now is, again, we start with a visual cue, either pointing to the left and right. 1/3 of a trial, we're going to have a switch. Oh, hey, just kidding, switch to the other side. And now we are specifically interested in this just kidding and see which part of the brain is more activated during this switch period. What we found is this right temporal parietal junction, and you can see the trace of the differential activation. It's much more activated in the switch trials compared to the whole trial, so the maintained trials. And there are other regions of the brain also, mainly in the right frontal gyrus and other regions, right frontal eye fields. More importantly, you can also look at the behavior, and the differential behavior in this task correlates with the activation of differential activation of RTPJ. Now, for those that are interested in autism research, you would also notice that RTPJ is very much talked about, and Maurizio Corbetta has a nice paper to talk about really RTPJ being more a shift, not only just shift in terms of perspective, but there's a shift in terms of location. So there's a broader way to interpret what RTPJ involvement in this situation. So now all this was done while I was still in Boston. So we decided to replicate our study and change it kind of when I arrived in Seattle. One of the questions that was raised in the last study was we were using visual cues. And so now we actually want to decouple the visual. Roland, I'm hearing a lot of feedback. Is there a way to not have that?
Let me see if this will help.
Okay. It helps. Can you still hear me? You can still hear me? So good?
We can still hear you, yes.
Okay, all right, great. So what we wanted to know is whether RTPJ, because RTPJ is being implicated in visual spatial attention, and we want to see, can we really isolate it to auditory spatial attention? And so now we have a cue right in the front of the trial, and we're going to tell you beforehand whether you should be switching attention for one stream of the sound versus the other. And so half the trials, you're going to, during the gap period, you're going to switch attention. So let's assume that we actually give you some cues to start off with, and you know which cue to listen to, which side to listen to. So for example, if it's a space trial, we're going to give you two sounds in the beginning to cue you to listen to, for example, the left side. In the whole trial, you're going to keep attending attention across the gap period. But if it's a switch trial, you're going to switch from EEM and then switch across to EML. Now we can cue you in this paradigm to only give you space cues. And so you're switching spatial attention in the middle of the trial. Or we can actually collapse the spatial features and have a high pitch and a low pitch and ask you to pay attention to one pitch feature and then switch to another pitch. And so the question we're asking is switching attention in audition the same if you're switching in space versus switching in pitch cues? So what we found is that when you, again, now it's only switching in spatial cues, we replicate what we found before, exactly the RTPJ cluster that is involved. And the timing is different because now you can anticipate when the gap is. And so the activation deviates even before the onset of the gap, because you can predict the timing. But when you're doing a pitch cue trial, it turns out that you use the LIPSP lateral inferior parietal sulcus. It really doesn't matter in this case. It's almost like the left side of the TPJ area. But again, it's only when you're maintaining, it's almost like you're humming the pitch to carry across the gap. And so the whole trial actually, it's more involved on this side. Now remember, we're using this to contrast space versus non-space. So what I want to sort of do a comparison is Marissa Corbetta has a really nice paper on summary of the top-down and bottom-up features of visual attention. And normally in vision, it's very space dominant in terms of the investigation. What we have found are regions that has the same regions for auditory attentional networks. So the left frontal eye fields, RTPJ, right frontal eye fields, right medial frontal gyrus, etc. But that's only true in the spatial auditory spatial tasks. When we talk about non-spatial tasks, it's actually getting regions that is not in this network. So essentially what we're really interested to know is how auditory attention is coupled to the oculomotor network. Now, Marissa Corbetta is talking about this visual attentional network, but really all these regions, FEF, IPS, are very much coupled to the oculomotor circuitry. So let's review the oculomotor circuitry a tiny bit. Now remember, we have the visual area projecting down to the superior colliculus. Superior colliculus is an interesting region. It actually encodes the vector of your saccade, where you want to point your eyes to, right? So, SC is crucial for saccade execution. And of course, the visual processing goes up to IPS. IPS also sends signals to move your eyes. And that eventually gets up to frontal eye fields. Frontal eye fields also communicates to superior colliculus for planned voluntary saccades as well. Now, if you pick up an auditory pathway textbook, normally you start from the cochlear, you have a lot of pathways, you go from the midbrain and the thalamus, and it inevitably stops in the auditory cortex. Little known fact that the secondary auditory areas has direct connections to the frontal eye field. So our question is, well, if frontal eye field is it's involved in auditory spatial attention, how far down does it go? Now, remember, spatial attention it's a processed entity, right? Because we have to calculate interaural timing differences. That's done in the olivary complex. Now, the auditory midbrain then projects to the inferior colliculus. That all feeds up to the auditory cortex. The superior colliculus and inferior colliculus is connected through the brachium. And a lot of the sort of cross-modal coupling and spatial encoding, whether it's head-centric, eye-centric, it's implicated around these areas. So it leads us to basically ask even more fundamental question, does the oculomotor circuitry influence auditory attention in a behavioral paradigm? We've sort of seen frontal eye field is involved in auditory spatial attention, but can it affect behavior? So in order to ask this, we basically came up with the question of, does gaze, moving your eyes, improve binaural acuity, right? Given the last diagram that you see from the connection all the way back down, could it improve your binaural encoding? So in this paradigm, what we did, it's done, Ross Maddox led this research. Now he's at Rochester, just got tenured actually. And so he asked the question this way. So we give you a primer, and then we're going to ask, the primer contains timing information and also selective information that I'll describe below. But the crux of the question is, we have two probes. One is slightly on the left and slightly on the right. And your response is, did the sound move to the right or did the sound move to the left? How we're going to have the primer is as follows. We have eye tracker, and we have the video tracking your eyes, and we will tell you whether the sound is coming from 0 degrees or 25 plus or minus 25 degrees. And we either give you that cue visually and ask you to move your eyes to land on that spot before we deliver the probe, therefore actually ensuring that you moved your eyes. We also have an auditory primer. So we test this with an ITD difference or an ILD only cue. So we give you the orthogonal cue as the auditory primer. So here's a burst of noise coming on the plus 25 degrees. Listen to that side. So we prime you for the auditory attention, but we don't do anything to your eyes. We also have uninformative primer. So we have a video on and just giving you no information, or even we just have a sound in the middle and don't give you any visual information. Or the uninformative primer basically gives you the same timing information, but no space information. We use eye tracker to make sure that we only deliver the probe at the time that's appropriate. Either you've moved your eyes explicitly to those regions, or that you're actually keeping your gaze at around 0 degrees when you're not supposed to move your eyes. Okay, so what happens in this investigation? Well, first of all, we know that for the longest time, almost for a century, that your acuity to auditory space is much better when you're in the midline compared to when you're on the side. And so we replicated this. You actually see a better discrimination in the center compared to the side. What is surprising to us is that given all the studies we've done before, when we prime you to space, you can segregate sound better. We assume that directing you the auditory attention in space would improve this task. What we found is that no, it did not improve the task, your acuity at all. What is surprising though is that when we moved your eyes explicitly, your improvement is there, and this is for the ILD difference, and it's also the same in the ITD difference. So what this tells us is that actually the oculomotor circuitry is really coupled to auditory spatial processing. Moving your eyes actually improve binaural acuity, and that's a pretty neat thing that that from a neuroimaging perspective leading back to behavior. And essentially gaze helps, but auditory attention surprisingly does not. Now, this first foray in terms of getting into visual space, and of course the more general question to ask is how do auditory and visual objects are interacting in any complex environment. We normally have both auditory and visual stimuli in a complex environment. So one theory to try to link these two modalities together is something called a temporal coherence theory. It posits that, at least in sound, all the different cues coming from the same talker, so the pitch cue, the spatial cues, the timbre cues, all co-modulated because it's coming from the same talker. That temporal coherence of that cue across frequencies gives you a way to bind to the same object. And so that was laid out by Shyam Sharma and colleagues back in 2011. And what we're interested in is whether you can extend this temporal coherence theory into cross-modal object. So let's assume that you can you have visual objects that co-modulates with sounds, not unlike when I'm speaking and the sound co-modulates with my mouth. Can that co-modulation create some binding element such that you can really have a particular feature here, the amplitude of the sound and the size of the mouth or size of the circle that we eventually end up using, can bind these objects together so into a cross-modal object. So we tested 3 cases. So here is a visual ring that modulates either coherently with the target, coherently with the masker, or not match at all in the middle. So intuitively, you would think that if the ring modulation is temporally coherent with the amplitude of the target, with the sound that you're supposed to be attending to, that should increase your performance. If I'm really evil and really want to make the task hard, I can have the visual modulating to the thing that you're supposed to be ignoring in the masker, and your performance should go down. And when it matches neither, it should be in between. And so this is really the question that we're asking is there a cross-modal objecthood? So the detail of this task, we have orthogonal amplitude modulation of a narrowband noise. So it's an amplitude modulation, but these two modulations are orthogonal. What we ask is whether the subject can report a target event. These target events can happen in the target and the masker, but you only have to report in the target and ignore the target event in the masker. Now, a target event, we did this in 2 sites, actually. We did it in Seattle with a frequency modulation, and in fact, they did this in Oxford with our collaborator, Jenny Beasley, and they had a vowel temporal shift. And so what happens is that the ring modulates either coherently with the target, with the masker, or neither. And just to keep the subject honest, we changed the color of the ring really briefly and their visual catch trial so that you actually have to attend to the visual stimulus as well. Otherwise, you can do this task without washing the rings, and we wouldn't know that you're not doing cross-modal task. So what do we find? So when the visual ring is co-modulated with the auditory amplitude, they did quite well, and you see a marked significantly decrement in the D prime when the visual ring is co-modulated with the masker. And you see somewhere in between for the match neither condition, as we actually predicted. So this is telling us that their modulation across time is actually pretty a good cue to really gel these two modalities together and help you in scene analysis. So we went one step further, and so Jenny Beasley did these recording in the ferret brain in the auditory area, and now she presented them with either this is always coherent, so the visual envelope is the same as the first auditory stimuli, and the visual stimuli is congruent to the second stimuli. But when she presents both the auditory mixture, so A1 and A2, two stimuli mixed together, combined with visual stimuli that is co-modulated with the A1 stimulus or co-modulated with the V2 stimulus, you see the auditory cortex actually segregates that sound back to the A1 from the mixture of A1, 2, or if you modulate with the V2 envelope, it pulls out the A2 in the mixture of A1 plus A2 sounds. And what you actually see is that this is true for both awake prep and anesthetized prep. So it's interesting that even for anesthetized animals, the visual coherence with the sound actually helps you segregate out a complex sound when they're in a sound mixture. And this got us thinking more about the theoretical underpinning of cross-modal objecthood. How do we establish that there's cross-modal binding? And if you're in the field of audiovisual research, binding is a very loose term that's used. Sometimes it means integration, sometimes binding, it's more perceptual. And we really want to segregate out these terms and really want to look at binding specifically when there's coherent across these objects. We also think that they can be bidirectional. So here, visual helps you in auditory task, and we actually think that the auditory binding, auditory coherence can actually help you with visual as well. So that's yet to be tested. Okay, so I talked about how went from scene analysis, trying to segregate sounds in a mixture, to space versus non-spatial elements that dovetails the cortical networks of vision. That led us to think about oculomotor circuitry, because moving your eyes also changes visual attention, and it turns out there's some link even to behavior, how the oculomotor circuitry is coupled with audition. And so now we want to take one step forward and ask, how are these areas connected? So we're interested in the functional connectivity analysis, and especially when people report that they can't hear sound properly or segregate sound properly in noisy environments. Turns out more and more we know that people can have typical hearing or audiograms, but they struggle in these noisy environments. And we posit that that may be because some of the functional connectivity is different, being from the neurotypicals. And so this work is in collaboration with Emily Fox now in Stanford University, Nick and Jordan Drew, so now graduate student. And we take a very machine learning engineering approach. We treat this as a time varying state-based system. And I'm going to just rush this forward a tiny bit. For those that are interested in this, we are looking at the innovation step in a state space modeling. The nice thing is that instead of looking at this as 5,000, 6,000 dipoles when we're doing inverse imaging, in this modeling, we end up having these pastellated areas that takes care of the MRI morphology, because the matrix it incorporates a forward matrix. and the noise levels it's low rank, and so it's nicely behavior such that we can actually do these type of connectivity analysis approach. So let me share some preliminary results. The cool thing is that there's no morphing over subjects, where we get a lot of inter-subject variance, and also we can make ROI specific inference. So what we did was pull out the ROIs that we talked about before. So auditory cortices, premotor, so frontal eye fields, visual IPS, LIPSP, RTPJ, all these areas that we've talked about. What I didn't show you is that we also implicate dorsolateral prefrontal cortex when we have even more complex of switching, when you're switching between cues, not just within the same cue. And now we subject this into the connectivity analysis. Now, so the way to read this in very complex slides, basically we're testing the deviation of that A matrix, the change matrix, and how far away that's away from zero. The further away it is from zero, it is an implication of that there's connectivity. Here it means this bilateral connectivity, both from the green condition and the blue condition. And we also have directionality. So here we can actually show that for this particular analysis, IPS leads auditory right cortex, for example. So just to summarize our preliminary data, remember, this is the cortical network that we're investigating, and now we really want to see how they connect up while you're doing an auditory attention task. So let's look at while you're doing a spatial task. When you're maintaining attention, it turns out we find these bilateral connections. Bilateral connections in the auditory cortex, bilateral connections in IPS, and also the frontal eye fields. Now, you would say, hey, you're doing an auditory task, bilateral connections in the auditory cortex, of course is going to be there. The interesting thing is we, again, introduce this gap period, and so it's maintaining attention during a gap when there's no sound, right? So the connectivity still holds when there's no sound, no bottom-up stimulus to drive that bilateral connections. Some other things that we can see, this flow of information, we included dorsal anterior cingulate cortex. So the ACC project information down to the auditory cortex for some controlling information. And they're also, from a higher order DLPFC, going to the frontal eye fields and communications between IPS and RTPJ, which is very similar to Cobbetter's diagram. And of course, the DLPFC communicates with ACC and also other areas, right? These communications happens bilaterally. Okay. What is really of interest to us is look at these type of connections and ask, when you have neurotypicals compared to autistic subjects. So we've just also come up with papers where we really talk about how autistic kids in classrooms struggle, even though they have neurotypicals, typical hearing profiles, that they struggle to pay attention to sounds, right? So they're either hypersensitive or hyposensitive, and that's documented quite widely in terms of this observation. And so what we're interested in is see whether there are differences in functional connectivity. These are the preliminary results. We have not enough subjects to publish them yet, but you see sort of recapitulations of the same network, both in the neurotypicals and ASD, which is a great thing, even just from a methodological standpoint. This seems like it's a poor man's version of a Sandy check, right? You're doing a check on your connectivity analysis. But importantly, you see the differences now slowly to emerge. So while there are more bilateral connectivity in the neurotypicals, these are missing in the ASD samples that we have. And so we really want to further investigate these type of differences, and there's some compensatory connectivity that you see in the ASD. And so here it's sort of really talking about neurodiversity, how they try to have different ways of connectivity, maybe to compensate for the typical brain network. Interestingly, we also have a sample from fetal alcohol syndrome disorder. So folks with FASD, in the first trimester of gestation, mother have alcohol, and that actually changes the frontal growth of the morphology. And we really thinking of FASD, ASD are different ways to investigate the auditory attentional network. And so things that moving forward, we really want to look at what happens to the connectivity when you're attending to space versus, again, pitch or other non-spatial cues. Do you wire up differently? And as I mentioned, the neurodiverse population is another window for us to look at how these connectivities are important in the attentional network and their behavioral manifestation. As I said, ASD listeners need more cues for selective attention in complex environments, and we know that we need to scaffold them with ways to listen better in complex environments. And that is also true listening difficulties in the absence of hearing loss. It's also very prevalent across FASD children. So those are the things that we're looking at moving forward. And now Bonnie Lau over at Oberlin Garge at UW, she's done tasks that for any of the audiologists here would know. It's basically the CRM corpus, coordinate response measure. It's a simple auditory task where you give them 3 streams of sounds, and you're only reporting one. It's widely used in auditory experiments, in psychoacoustic experiments. But what she found is that there's a huge correlation between IQ score, the WASI-2, with what we psychoacousticians think that it's just a pure task to assess auditory processing. And it turns out that a lot of cognitive factors really change how the measurements that we traditionally think as just pure psychoacoustics measurements. I'm going to leave some time for questions, and just want to acknowledge past fundings that enabled us to do all this work. And also, now that I'm chair, I feel like I'm more like this person more than the situation down here. And for those that can actually tell me offline what this title actually, what I have blanked out, I will probably buy you a drink afterwards. So thank you for your attention.
Thank you very much, Adrian KC. That was really nice talk. 1 second here. I'm unmuted on my laptop, so it's okay. But right. Sorry, 1 second, Adrian.
No problems.
Always some problems. Okay, well, I'm dealing with unfortunate computer issues. Maybe I'll just use this to ask a quick question about your source modeling that you were talking about early on. So you, what did you mention? You mentioned a penalty term. I was wondering if that was a sort of normalization, regularization term that's normally used, or if you're doing something special there because this is an auditory sources.
Yeah, no, we don't. We actually use the L2 norm. So let me bring back that slide.
Right.
I think what you're really referring to is this penalty term.
You mentioned a penalty term. I was wondering if that was a
No, we use the L2 normalization, which leads to a nice linear algebra inverse expression. Of course, you can use L1 where your solution is much more punctate. And of course, now there is the L1, L2 mixed norm, right? So take into account the spatial specificity while using the L2 relaxation across time. We use the most used arguably is the L2 norm as the penalty term. So our sources are necessarily broad because of the L2 penalty term, right? Because L1 has a more punctate spread, but that's part of the things that we have to live with inverse imaging.
Okay. Thank you, KC. So still just having some computer issues on our side, but we are still all here. Does anyone here in the audience have questions? Yes, Erica. That might be helpful for the moment until we get this back on. Wait, turn on the video.
Hi, Adrian. I'm Erica Skoy. I don't think we've had a chance to meet, although I think we run in some of the same circles.
Yes, hi.
Nice to finally hear a talk from you.
Yes.
I'm going to ask a little bit of an off-the-wall question, but I have my kind of hearing scientist hat on right now. I'm curious what you know about age-related changes to the oculomotor system and the degree to which they may parallel age-related changes to the auditory system and the implications that that might have for spatial listening, especially in somebody who is about to receive a hearing aid or has just received a hearing aid.
I would really want to know more about it. I'll be honest with you, we put in a grant a while back to look at sort of the hearing loss that look at all these cortical areas. I think now wearing my sort of AUD study section hat and where people are at this, most still think that the cortex really doesn't change as much as the peripheral deficit, and it's the peripheral differences that lead to upstream differences. But I'm absolutely with you in terms of really looking at aging, how that relates to hearing. Sometimes hearing scientists are too siloed to really look at broader how aging affects the rest of the cortex. And I think you're absolutely spot on. That really needs to be investigated. And unfortunately, I think their work coming from G.D. Dubnow's lab, right, to really look at that, but they're not that broad investigation. And I think, yes, we really need to look beyond the potential peripheral deficit. It could be if there's a feed forward and feedback differences, right? Now that you have poorer temporal information and the ITDQ is worse, and somehow your oculomotor circuitry is also weakened because of aging, and then you have a double whammy, right?
Your talk made me think of something that I've certainly heard people say, and I've actually said myself, which is, hold on a second, I can't hear you. Let me grab my glasses.
Oh, yeah, that there are many instances that when I first thought about frontal eye fields, it didn't really, it's not, because I think it's our upbringing, right? When we have auditory classes, we don't really think of it this way, but sometimes when you close your eyes, you can hear this clocking sound of the high heels walking across the corridor, and you almost close your eyes and think that you can move your eyes to locate that sound. That to me, there's a lot of these frontal eye fields moving your eyes to tune into space. It's actually more happening. Since you're asking sort of a more, you phrase it as wild topic. I don't think it is, but I'll give you a wild conjecture. The dorsal cochlear nucleus. It's important for moving the pinner in cats, and it's a lot of the cross-modal coupling for spatial hearing, and interacting with the external world. Somehow I think that frontal eye field is actually driving to the DCN, except we can't move our ears. And there's the eye-centric and head-centric movement that we have to resolve. And so I refer you to Jenny Grove's work, and I think it's plenty fascinating in terms of where that approach is, in terms of spatial hearing and how that relates to the cortex.
Yeah. Online audience. All right, looks like you may have a rather short question.
Sure.
So much, Dr. Lee.
Thank you.